how can a headache pill start working in what feels like minutes, but other meds take hours or sometimes even days to really kick in? Well, the secret isn't just about how fast you absorb it. It's all about distribution, how that drug travels from your blood out to the tissues where it actually needs to do its job. So, here's our game plan for today. We're going to break this down into 5 key parts. First, we'll see how the journey begins. Then, we'll cover the rules of the road that every drug has to follow. We'll navigate some high-security zones in the body. After that, we'll learn how we map this journey using a really important value called VD. And finally, we'll see how this all has a massive clinical impact. Okay, so the journey kicks off the second that drug hits the bloodstream. Distribution is pretty much the movement of a drug from the blood, our central highway, out into all the different tissues and fluids of the body. And, key point, this is a two-way street. It's a reversible process. To really get this, you have to think of the body as being, well, mostly water. For a typical 70-kilogram person, that's about 42 liters of water. The drug's journey starts in this tiny little 3-liter puddle of plasma. And from this central hub, it has to find its way out into the fluid that surrounds our cells, and then, hopefully, inside the cells themselves. And if we zoom way in, down to the micro level, you can see what's happening. The drug molecules, and this is important, only the free, unbound ones, squeeze out of these tiny blood capillaries. They enter the interstitial fluid, and from there, they can move into the tissue cells to finally get to work. I really want you to lock in on those 2 ideas, free drug and reversible. The drug is constantly moving back and forth, trying to find a balance. Okay, so this whole trip isn't just a free-for-all. It's actually governed by a pretty strict set of rules of the road, if you will. And these rules decide where a drug can go, how fast it gets there, and how much of it actually arrives at the final destination. We can basically boil these rules down to 3 main factors. First up, tissue permeability. Does the drug have the right passport to get through the door? Second, perfusion rate. Is it cruising on a super highway with tons of blood flow, or is it stuck on a slow country road? And number 3, and this is a big one for your exams, protein binding. Is the drug actually free to do its job, or is it tied up and basically inactive? Let's break these down. So this first rule, permeability, is all about the drug's passport. Think about it. Our cell membranes are basically made of fat, or lipids. So drugs that are also fat-loving, we call them lipophilic, and have no electrical charge, they can just slide right through. But drugs that are polar or have a charge get bounced right off that fatty barrier. It all comes down to their chemical properties, like pKa and the pH of their environment. Next up is perfusion, which is just a fancy word for blood flow. Imagine it's a delivery network. Organs with tons of blood flow, like your kidneys, your liver, your brain, they're the superhighways. They get flooded with the drug almost instantly. But tissues with poor blood flow, like fat or bone, they're the slow backroads. It takes way longer for the drug to get there, and that directly impacts how fast it starts working. All right, but some parts of the body are like high-security zones, real VIP areas. They have these extra physiological barriers that are super selective about what they let in, acting like tightly controlled checkpoints. And the most famous one, of course, is the blood-brain barrier, or the BBB. The cells here are fused together by what we call tight junctions. They form this nearly solid wall that protects our central nervous system. This means only very specific, usually very fat-soluble drugs can get a pass to enter the brain. Then you've got another super important one, the placental barrier. This separates the mother's circulation from the baby's. Now, while it does offer some protection, it's way less strict than the BBB. A lot of drugs can actually cross it. And this is exactly why we have to be so incredibly careful with medications during pregnancy to avoid any harm to the fetus. Okay, so we've got all these rules, all these barriers. How in the world do we actually measure this? How do we map out a drug's journey? Well, for that, we use a really cool concept called the volume of distribution. Now, this is super important. You got to wrap your head around this. The volume of distribution, or VD, is not a real physical volume. It's a hypothetical number, a theoretical concept. It's a way for us to figure out the extent of a drug's distribution. Did it stay in the blood, or did it spread out far and wide into the tissues? The math is actually pretty simple. VD is just the total amount of the drug you put in the body divided by the concentration of the drug you measure in the plasma a little later. But this simple little ratio tells us a whole story about where that drug decided to hang out. So what's the story it tells? Well, if you calculate a low VD, something like 3 to 5 liters, that means the drug concentration in the plasma is high, so the drug mostly just stayed put in the blood. But if you get a high VD, and I mean sometimes hundreds of liters, way more water than is even in your body, that tells you the plasma concentration is super low. Why? Because the drug has left the blood and is now hiding out deep in the body's tissue.
Okay, let's bring this all home. We're going to talk about the clinical side of things, specifically the huge impact of protein binding. This is where the textbook stuff meets real patients, and it's absolutely critical to understand. If you remember one thing from this, make it this. Only the unbound, the free drug is active. A drug molecule that's stuck to a protein, like albumin in your blood, is inactive. It's just a passenger. It's not the driver. Only the free portion can actually cross membranes, find its target, and cause an effect. Now, drugs can bind in two main places. You've got plasma protein binding, which is usually a temporary, reversible thing. It acts like a reservoir, keeping the drug in the blood and giving it a small VD. But then there's tissue binding. This can be much stronger, sometimes even irreversible. It pulls the drug out of the blood and locks it into tissues, leading to a huge VD and often a much longer half-life. Okay, but this is where it can get really, really sketchy. What happens if a patient takes a second drug that fights for the same parking spot on that plasma protein? This is called displacement, and for certain drugs, a tiny bit of displacement can cause a massive problem. So just look at these numbers for a second. Let's take drug B. It's 90% bound to protein, so 10% is free and active. If another drug kicks off just 1% of it, the free amount goes from 10% to 11%. That's a 10% increase. Not a huge deal. But now look at drug A. This is the crucial part. It's 99% bound, leaving only a tiny 1% free to do the work. If we displace that same 1%, the free concentration goes from 1% to 2%. It doubles. That is a 100%, a doubling of the active drug in the system. You have effectively doubled the dose the patient is seeing without them ever taking another pill. And this is a classic, high-yield reason for dangerous drug interactions and toxicity. And that sudden spike of free drug, it just slams the body. The newly freed up drug rushes to its target, causing a much stronger effect, which can often be toxic. It also completely changes how the body gets rid of the drug, because now there's way more available for the liver and kidneys to process. So let's end with a clinical thought experiment. We know the liver is the factory that makes most of our plasma proteins, like albumin. So, if you have a patient with liver failure and their protein factory is shut down, what do you think is going to happen to the effect of one of those highly protein-bound drugs? Understanding distribution gives you the answer.